Hello, everyone. Thanks so much for joining us for another webinar organized by the IWGC. Today it's my pleasure to have Dr. Kenta Shirasawa. I will briefly read his bio here, and then we can keep going. Dr. Kenta Shirasawa is the head of the Laboratory of Plant Genetics and Genomics at the Kazusa DNA Research Institute in Japan. He holds a Ph.D. in agricultural science from Tohoku University. He specialized in utilizing advanced genomic technologies and bioinformatics tools to analyze understanding and the complex genetic architecture composition of more than 50 plant species. In addition, Dr. Shirasawa has organized the Kazusa Genome Project aimed at sequencing genomes across all orders of angiosperms at the telomere-to-telomere level to understand the phylogenetic relationship of angiosperms from the aspect of genomic structure. So, Dr. Shirasawa, it's my pleasure to have you here, and I'm looking forward to your presentation today.
Okay, so thank you for your kind introduction, and thanks for inviting me this seminar webinars. So today's my talk is about T2T genome project across all orders of the angiosperms. So T2T means telomere to telomere. Telomere is a component at the end of chromosome, both ends of the chromosomes. So T2T means one telomere to other telomere. We could sequence, we could determine the nucleotide sequences of the chromosomes. So using these technologies, we are now trying to determine the genome sequences of all orders of the angiosperms. So first, I'd like to introduce our institute. Our institute name is Kazusa DNA Research Institute. The building you can see here. And our institute is located in Chiba Prefecture. Chiba is a name of prefecture near Tokyo here. And our institute has two departments. One is Department of Applied Genomics, and the other is Department of Frontier Research and Development. So our laboratory is located in this site, Frontier Research and Development, and our name is Laboratory of Plant Genetics and Genomics. So the mission of our laboratory is to sequence the genomes of plants and analysis to develop breeding tools to improve crop, for example, crop productions or crop resistance to disease, or improving the quality of the products. So using the genome information, genome technologies, we try to contribute to the industry. So here is our kind of history of plant genome sequencing in our lab. So we started with Arabidopsis in 2000. You can see here. After that, we sequenced more than 50 or 60 plant species, shown here. So using different sequencing technologies like Sanger method, 454, Illumina, and now we are using PacBio read technologies. So anyway, using these technologies, we determined the genome sequences for these crops, because our mission is contributing breeding, improving the crops. So that's why we selected crops as a target for this sequencing analysis. So today's my talk contents shown here. I have three topics. So the first is the T2T Genome Project across all orders of angiosperms. The second is the T2T Genome Assembly for allotetraploid pernicious with echinochlora phylogons. The last is the leaf color evolutions by urban heat stress in Oxalis colorata. So I started with the first one. So I'm not sure the audience are familiar with the genome sequencing. So I started with this slide, the definition of the genome sequencing. The old or initial period of the genome sequencing, we generated draft sequence of the genome. So draft means not complete. For example, you can see here the yellow bar. This indicates a chromosome, and black boxes indicate repeat sequences. Repeat means a transposon or retrotransposon, multi-copy sequences in chromosome. So anyway, we try to determine the sequence of this chromosome. At the initial stage, we could use short-read technologies like Illumina or 454. So using the technologies, we could generate data for the sequence reads for this chromosome. The data including some yellow part and gray part. Anyway, using these sequence data, we could assemble, we could construct the sequence of the chromosome. However, sometimes the sequence are stopped, assemblies are stopped at this point. Means transposon sequences are large copies in the chromosome. It was impossible to connect here and here because this read we don't know the information. This is where this reads are coming from. So this single chromosome were assembled, constructed into 4 pieces. You can see here. So we call them contigs. So in this picture, one chromosome assembled into 4 contigs. However, we don't know the location of contigs on the chromosome. So after that, we could establish chromosome level or chromosome scale sequencing with other technologies like genetic mapping, optical mapping, or high-C. So these technologies provide the information of the orders of the contigs. So like this, we could obtain the information of the orders of these 4 contigs like this. But the sequence, nucleotide sequence between contigs were not determined. So we need to connect, we could connect contigs with N. N is a represent undetermined sequences. Anyway, we could establish chromosome level or chromosome scale sequence using short read and genetic mapping, optical mapping, or high-C. So now we can obtain many types of chromosome level sequences for many plant species. So we summarize the chromosome level sequences for more than 100 plant species. So you can see here, the white bar indicates sequences, IWGC sequences of the chromosome chromosome level sequences. The dot line indicate the chromosome length. However, most of the case, the chromosome level sequences not reach to the chromosome length because of the gap sequences, gap or undetermined sequences there. So, however, we call them chromosome level sequences. So I don't think actual sequence data for the chromosome are not available from the chromosome level sequences. So after that, we could establish telomere-to-telomere T2T sequences with new technologies like long reads. Long reads can generate this lead. This lead can cover completely cover the repeatable sequences in the chromosome. So these leads can useful to generate to assemble config. This config includes completely includes the sequence of the chromosome. So we call this config as telomere-to-telomere configs or telomere T2T sequences. So this is the final goal of the genome assembly representing the entire complete sequence of a chromosome. So to achieve this T2T assembly, we installed the PacFile sequencers last year, and we are now mainly using this machine to generate T2T assembly for several plant species. So in addition to the sequencing technologies, we are using high-C or omni-C technology to scaffold to extend the contigs into chromosome level, or optical mapping also used. And another techniques is genetic mapping. This is a classical method, but very powerful approach to generate telomere-to-telomere assembly. So sometimes, in many cases, for genetic mapping, mapping populations are required. However, some plants, it's difficult to generate mapping population. For example, you can see here, this plant is a family of brassica. They have a self-incompatibility, so it's impossible to obtain self-mapping population. Or other case of trees, it's also difficult to generate mapping population because of the long lifetime. So for the species, we developed a new mapping techniques using single pollen, single sequencing technologies. So, pollen has a haploid genome, and each pollen has different haploid genomes. Means each haploid genome has a recombinance of parental two chromosomes. And the recombinant point in the genomes in pollen are different depending on the pollen. So we could recognize these pollens mapping populations if we could genotype one pollen by one. So to achieve this idea, we sorted the pollen one by one using the single cell sorting technologies. And after that, we could genotype the SNP in each pollen by transcriptome data. After that, we could analyze the linkage of the SNPs to make genetic map like this. Then we could merge this genetic map with the context from the sequencers to generate chromosome scale or T2T genome assemblies. Anyway, using the long-read sequencing technologies and IC or optical mapping or genetic mapping, we now trying to make a T2T genome assemblies for many plant species. So why we started to telomere-to-telomere assembly for plant species, I would like explain that one of the reasons in this slide. So when we determine the genome sequence of Japanese chestnut, we found some interesting points. So when we compared the genome structure of Japanese chestnut with Chinese chestnut, the chromosome structure was conserved, completely conserved between two species because two species are very closely related. So when we compared Japanese chestnut genome with peach, we could detect some synthetic regions like here. If when we compared Japanese chestnuts with cacao, we could detect synthesis like here. Versus grape, cassava, and maple, in all cases, we could detect synteny or genome structure similarity between Japanese chestnuts and these species. So we thought the relationships of Japanese chestnuts and others are very close. However, in the phylogenetic trees, Japanese chestnuts is actually close to the peach. However, other species are not so close. And Japanese chestnuts is most closest to orders of cucumbers. However, the genome structure between chestnuts and cucumbers are not conserved. So we thought the genome structure is not reflect the relationship in this phylogenetic trees. Actually, this tree is drawn based on gene sequences of plasmid. So we are thinking the plasmid gene evolution and genome structure evolutions should be different. So that's why we started try to understand the genome structure evolutions using the T2T genome assemblies. So we launched a project, namely Kazusa Genome Project, to sequence the orders, because here is a big tree indicating angiosperms, and this tree included 60 orders here. However, some of the species, around 20 species, no genome information are available. Like this red mark indicate no genome information are available. So this moment, it's impossible to compare the genome structures of all orders of the angiosperms. So we are now targeting these red marked 20 species for T2T genome assembly. So this is the status of the assembly result. We collected these plant species or plant orders from the collaborators to determine the genomes at the T2T levels. So it was very interesting. It's a very interesting project because the genome size of the orders are very different. For example, this lily has the biggest genome, like 40 gig genome, so huge. And the assembly results are maybe good, but some of the plant order, we could generate T2T already. However, some of them, it's difficult to make a T2T assembly, only the sequence, long read sequencing technologies. So we are now trying to generate T2T assemblies using other techniques like IC or genetic mapping. So in this activities, we could establish the T2T assemblies for several species. So today's topic number 2, I'd like to introduce our attempt to assemble the allotetraploid pernicious weed, namely Echinoclora pyrophobon. So this plant is a weed in paddy fields, making yield loss. So this weed controls are performed by a herbicide. However, sometimes herbicide resistant mutants were found in the field. So this is a big problem to agriculture. To to identify or to understand the genetic mechanism for this herbicide resistance. So the Dr. Iwakami in Tokyo University of Agriculture and Technology invited us for this project. First, we need to sequence the genome of a plant species. So this genome is a little bit challenging to the genome project because they have allotetraploids. Allotetraploids means they have two genomes from two different species. And the chromosome number was not so big, 32N equals 36, and the genome size is around 1 kilobase. Actually, this genome information was already published when we started this project. However, the completeness is not enough to cover the entire genome. So we started this genome sequencing project with the long read sequencing technologies. So we used line from California, namely R511, for genome assemblies. So high-fidelity sequencing obtained this line for genome assembly. In addition to the line, we developed a mapping population from a cross between R511 and S401. So this mapping population was genotyped with DD-RAD-seq technology for SNP linkage analysis. So this is a result of the assembly of the long reads. So actually, we obtained 70x coverage of high-fi reads for this line. And however, the assembly results were not so good. So we wanted to improve this assembly. So we tried downsampling. That means only 45x coverage data are subsampled from this 70x reads. So using this 45x reads, the assembly result was very improved. That means some of contexts were connected in this 45x assembly. So finally, we could determine we could obtain long contigs for this species. After that, we developed a genetic map based on the mapping populations. Like here, we could establish a genetic map, and on this linkage map, we could align the contigs to connect here to establish a chromosome length contigs. So this is a resultant assembly of this species. Sorry, this is not 12. Actually, this is 18. Anyway, 12 is. This species has 18 chromosomes. Among them, 12 chromosomes are assembled at T2T level without any gaps. So you can see here, red bar indicates T2T assemblies. And we could assign these 18 chromosomes to relative to identify the genome. That means half of 18 chromosomes should be A genome, and the other should be B genome. The cross chromosome, we named them A genome, and the distant chromosome are named as B genome, like this. So using this information, we compare the structure of genomes between our target species versus relative species and previous version of this species. As you can see here, the continuous line indicates that the genome structures of this species are highly conserved between close relatives and previous version. So we confirmed the quality or accuracy of the current genome assembly was high. Of course, because we could sequence entire regions of the genome, we could find some differences between the previous version and the current version. You can see here and here, these regions are sequenced or assembled in this study because they are lacked in the previous version. And actually, this region are enriched with repeat sequences. So this result suggests this region should be centromere or pericentromere region. So anyway, we could establish a T2T level genome assembly for this species, and we found the interesting point when we compared the A and B genomes. For genome size, we found the genome size of B genome is bigger than the A genome. The A genome size was 450 megabase. However, the size of B genome was more than 500. So why these differences are generated? So we focused on repeat sequence. So as expectedly, we could find the genome has enriched with some transposons like LTRs or DNA transposons and some unique repeat sequence of to this species. The most important point is the gene number. So gene number in B genome was less than that of A genome. So we thought the genes in B genomes were disrupted by the repeat sequences, like transposons are jumped into the genes in the B genomes. So this is our hypothesis. So based on this So we are now thinking the evolutionary model of this for this species. So actually, the target species are allotetraploid. So this will be generated by polymorphization between A genome ancestors and B genome ancestors. However, only in B genome ancestors, repeatable sequence shall be accumulated that cause gene extensions and gene disruptions. So this is our tentative evolution model for this species. Anyway, using this information, Dr. Iwakami is now trying to identify the mutant genes that produce with herbicide resistance in the field. So the last topic of this presentation is another weed species of Zaris. So we are now investigating about leaf color evolutions by urban heat stress observed in this oxalis. So this project is collaborated with Dr. Fukano in Chiba University, and I'm not sure the audience knows oxalis, but most of all may know this species. This species has a three leaflets shaping shaped leaves. As indicated here, the color of the leaves are variable. So some oxalis has green leaves, but some has red leaves. So we wanted to know why these color variations exist in this species. So Dr. Fukano investigated where the green leaves or red leaves are found. So we investigated the place, some place in Tokyo, and found the oxalis leaf color in green space. That means fields. We found only green color oxalis. However, in urban region means a city region, we found not only green color, but also red colors. So this observation this result were obtained from a small region of Tokyo, but it found some larger regions. And this result are observed actually in worldwide. So we thought this green and red leaves are results. This color variation are kind of results of the evolution of oxalis in different environment, like field and city. So why is the color variations are available in this oxalis? So we measured the anthocyanin contents in leaves, and we found the differences. So red color leaves contains the anthocyanin in leaves, like here. So red color red oxalis are found in roadside or some city region. They are red. They have red color, red leaves. Why they have red leaves? So we thought some city environment to adapt the city environment, the oxalis have to accumulate the anthocyanin. So one of the city condition could be heat stress. So we test the growth ratio of red and green oxalis under the heat stress. In control conditions, the growth of green oxalis is much bigger than the red ones. So natural condition or field condition, green plants are more active than red one. However, on the other hand, in city region, like heat stress conditions, the growth amount were bigger in red leaf oxalis than green one. So we concluded the accumulation of anthocyanin is a kind of adaptation to the urban environment for oxalis. So we thought that this adaptation is some evolution, like mutation should be happened in the genome. So we collect 100 oxalis plants from the Tokyo regions and analyze the SNPs to make these phylogenetic trees. So based on the result of this analysis, we found the red oxalis are joined to this cluster. That means the genetic variation is very low rather than other cluster for which green are mainly joined. And we thought the red oxalis are generated or evolved from a green line in this cluster. So we are now searching the causative gene to determine the leaf color. So for that, we assembled the genome oxalis genome at T2T level. Actually, oxalis is an octoploid. They have 8 genomes in cell, and this is the result of the red oxalis assembly and green oxalis. And when compared red and green, we found some conserved chromosome, like here, but some chromosomes in red were missed in green. So we are now thinking the red one is a arrow tetraploid, arrow octoploid, and green are odd octoploid. So arrow means they have different genomes in cell, but green odd octoploid has same genome, same 8 genome in cell. So this may be leading the variation of leaf color. So to identify the genes that control leaf color, we crossed red plants and green plants to generate mapping population. So the color was segregated in 3-to-1. That means the leaf color was controlled by a single dominant gene. And the gene position was identified in the genome. So now we are trying to identify the genes or cause of mutation in this point. That may explain how these oxalis evolved from the green. So once we could identify the genes or mutations, we want to try to expand this finding to across Japan, because we now studying only Tokyo oxalis, but we want to know this mutation can explain other city regions, or other city regions has independent evolution patterns. To test it, we need to collect Oxalis plants from all over Japan, but it's difficult for only us to collect so many samples. So now we are perform the citizen science for which citizen people can attend this project to understand the color variation or how occurred. So we are now asking people to send the Oxalis living in their places, and now we collected more than 1,700 plants from 70 sites for Japan. And the participant can attend our event, try to extract genome DNA from Oxalis, because many kids or some other participants of this event, it's a first trial for them to extract DNA. So this is a nice opportunity for them to understand what is DNA, what is genome. So if you have interest in this project, please access the website from this QR code. So because DNA or genome is a kind of famous word in this COVID-19, however, I want the people to know the DNA, genome, or PCR actually interesting or fun for to understand the citizen science. So that's why we started this citizen science project. So this is the conclusion of my presentation. So the T2T genome sequencing across all orders is ongoing with high-fi, ultra-long reads, high-C, and direct mapping technologies. So the genome information will be helpful to reveal biological questions, and some projects could be useful for outreach of citizen science. So finally, I'd like to thank contributors, Dr. Iwakami, Dr. Fukano, for the project presented in this slide, and I'd like to thank Dr. Sato, who is work on the weed species, and Dr. Ibra, he is working on Oxalis genome analysis. And all of the projects are supported by our lab members and field funding agencies. Thank you for your attention.
Awesome. Thank you so much. I appreciate it. I really liked all three projects that you covered. Very amazing. So if anyone attending the seminar would like to ask a question, there is a Q&A tool here at the bottom. Just go and write your question, and I will go ahead and read. I do have a couple of questions to get started. So can you talk a little bit about, I saw one of your species, well, a few of them in the lily family, the Lilium longiflorum, for instance, it has a genome of 40 gigabases. So can you talk a little bit about what are the main challenges with assembling a genome that large compared to some others that are like 1 gigabase, 40 times less amount of DNA content. So can you talk a little bit about what are the main, is this like a computational, with the amount of data, what's the main challenge?
Okay.
Yeah, that slide.
So the biggest genome was found in this lily. So the genome size was around 40 giga. So this is a big challenge for us. So, but actually genome sequencing and genome assemblies was very easy. And after assembly, we could establish already a T2T level genome assembly for this lily. This was also easy, but after that, there are some kind of problems to analyze the data. One of which is the size of chromosome. Actually, Lily has 12 chromosomes. Total length was 40 giga. So that means the biggest chromosome include 5 or 6 giga length. So this size is not suitable for the software tools. Actually, most of the software tools are designed for less than 2 giga. So many times we have the errors from the tools. So we need cut again this big chromosome into short pieces. This was a big problem for us.
Yeah, okay, that makes sense. That's interesting. Okay. And then you basically you divide the chromosome and then assemble them individually, the individual pieces. Okay.
Yes.
Interesting. And another question that I also have is can you talk a little bit about the kind of flow assembly? Because you said you downsized from 70x to 35x. So, usually we hear and we have the conception that the more data we have, the better. So does that mean there is an optimum actually and not always the more we have, the better?
You mean this result?
Yes, yes, yes. Yeah, you said for 70, the 45x was better than the 70x. Is that right?
Yes.
So
Yeah.
why is that?
Yeah, this is surprising for me. Yeah. Actually, in normal case, bigger data generate better result. But in this case, 70x coverage assembly didn't the best result. So we are not sure, but maybe some sequencing error in the release were accumulated in this 70x coverage. So that if so, the assembly are broken at the error point. So if we could decrease the coverage like 45x, the error were not duplicated in the data. So that means the content can connect correctly. So actually, we tried this downsampling 3 times. Means we tested 3 subsamples. We tried 3 sub-data sets to generate this T2T assembly. So
Okay.
downsampling is one of the solutions to make T2T assembly.
Mm-hmm. Okay, awesome. Interesting. Thanks for sharing that. And I have one final question. Can you correct me if I'm wrong, but one of the oxalis species, one of the oxalis ecotypes has one chromosome less than the other. Is that correct? I think your dot plot was showing.
This one?
Yeah, the red one has a chromosome that doesn't match the green. Is that right? So are they still compatible? The red and the green plants, they're still what about the progeny? Is the progeny fertile too? Okay.
Yeah, actually red and green are same species, and actually we observed the chromosome number in red and in green. The number are identical,
Okay.
but the assembly result were a little bit different.
Mhm.
So Now this is our assumption, but for example, red has a different genome, mean A and B genomes.
Mhm.
Green has only A genomes. In that case, red generates A and B assemblies, and green generates only A assemblies.
Mhm.
So if so, this picture is reasonable. We are not sure, but this is our hypothesis.
Mhm. Okay. Yeah, that makes sense. Awesome. So I think if there are no other questions here, I think I'm gonna do you have any final uh messages you would like to give, or yeah, well, thanks so much for your time. I appreciate it. Very nice to meet you, and we'll have our next webinar in a few weeks. So if you guys would like to join us, we'll be sending out some announcements sometime soon. Thank you, Kenta, again. I appreciate your time, and
Yeah,
have a
thank you
good
very
rest
much.
of your day.
Have a good day.
Bye bye.
Bye.
Bye, everyone.